Imagine a world where your child's eye color, height, intelligence, or resistance to disease isn't left to chance, but to choice. Not decades from now, but perhaps within our lifetime. Welcome to the frontier of genetic engineering, where the once science fiction concept of designer babies is quickly becoming a scientific possibility. But what exactly does it mean to design a baby? And what does the science actually allow us to do today? Let's start with the basics. At the core of this debate is something called CRISPR-Cas9, a gene editing tool that works like a pair of molecular scissors, capable of cutting DNA at specific points to insert, delete, or replace genetic material. It's precise, it's powerful, and it's already transforming how we treat genetic disorders. Originally developed for bacteria as a defense mechanism, CRISPR has been repurposed by scientists to edit genes in plants, animals, and yes, human embryos. In theory, CRISPR allows us to correct inherited diseases like cystic fibrosis, Tay-Sachs, or Huntington's before a child is even born. But there's a crucial distinction to understand. There's a difference between therapeutic editing and enhancement editing. One seeks to prevent suffering, the other aims to optimize. Preventing a fatal condition in an embryo may seem ethically sound, even noble. But choosing traits like height or intelligence? That's where things start to fracture along ethical lines. In 2018, a Chinese scientist named He Jiankui claimed to have created the world's first genetically edited babies, twin girls named Lulu and Nana. He edited their genomes to be resistant to HIV. The scientific community responded with outrage, not because he edited human embryos per se, but because he did it without consensus, oversight, or long-term safety data. He was later sentenced to prison in China. Still, the door had been opened. The idea of designer babies, once confined to speculative novels, had stepped into reality. And the world wasn't ready. But the potential hasn't gone away. In labs across the globe, scientists are quietly refining gene editing tools beyond CRISPR. Technologies like prime editing and base editing promise even more accuracy with fewer unintended mutations. The tools are getting sharper, and the temptation to use them is growing. So let's ask the question, if we can design better humans, should we? Supporters argue that we already design our children in less precise ways. We choose partners based on intelligence, physical traits, and personality, consciously or not. We send kids to better schools, give them nutrition plans, music lessons, tutoring, all to give them an edge. Isn't genetic editing just another tool in that long arc of parental ambition? On the other side, critics warn of a slippery slope toward genetic inequality. If only the wealthy can afford enhancements, we may create a caste system rooted not just in money, but in biology. This isn't paranoia. It's already visible in early consumer genetics markets. Some companies offer embryo screening, not editing, for traits like height potential or low risk of diabetes. It's legal, it's profitable, and it's mostly unregulated. So far, these screenings only select embryos based on existing genetic combinations. But as editing becomes viable, we enter the territory of customization, not selection. Let's talk about what's realistically possible. Right now, we can only edit simple, single-gene disorders with confidence. Diseases caused by a single genetic mutation, like sickle cell anemia, are clear targets. But traits like intelligence, athleticism, or musical ability are polygenic, influenced by hundreds, sometimes thousands of genes. We don't fully understand the interplay yet. Plus, genes don't act alone. Environment, upbringing, and randomness all shape who we become. Editing genes for high IQ, for instance, may not lead to genius. It could unintentionally disrupt other systems we don't yet grasp. That's the scientific limitation. But there's also the philosophical one. What does it mean to be human if our attributes can be programmed? Would children born through enhancement carry the burden of engineered expectations? And would natural births become an act of rebellion or of poverty? There's also the risk of homogenization. Imagine a world where everyone selects for similar traits: tall, athletic, high IQ, symmetrical features. Genetic diversity could collapse. Our quirks, our imperfections, our unpredictability might disappear. And with them, some of the very things that make us human. Still, some argue that this moment is inevitable. Technology rarely waits for consensus. IVF was once controversial. So was organ transplantation. Today they're routine. The same trajectory may apply to embryo editing. As safety improves and public sentiment shifts, what's fringe becomes familiar. In the future, nations that regulate too strictly may fall behind. Countries with looser laws might attract so-called fertility tourism, where parents travel abroad to access technologies unavailable at home. It's already happening with surrogacy and egg donation. Will gene editing follow the same path? Perhaps. But some scientists propose a global moratorium on heritable gene editing until there's international agreement, not to halt progress, but to guide it. They argue that once changes are made to an embryo's DNA, they're passed on to future generations. There's no undo button, no second chance. Others are less optimistic. They believe we've already crossed the ethical Rubicon. The question is no longer if designer babies will exist, but who gets to decide how. And here, perhaps, lies the most vital question of all. Can we build a world where this power is used wisely? Not for vanity, not for supremacy, but for genuine human flourishing? If we don't ask that question now, we may be left asking it too late, because the science is moving faster than our ethics. And one day soon, someone will walk into a lab, not to heal a disease, but to sculpt a legacy. And the child born from that choice will live in a world we helped shape, without ever choosing it.